Hi, my name is Roos Buitenhuis. I'm a research scientist in biological control at Vineland Research and Innovation Center. This video is an adaptation of several presentations about cutting dips I gave in 2018 and 2019. The making of this video was supported by funding from the Canadian Greenhouse Conference, and the project partners are the Vineland Research and Innovation Center, OMAFRA, Greenhouse Canada Magazine, and Flowers Canada Growers. Many ornamental plants are started from cuttings. Starting a crop with clean plant material is one of the most important factors in integrated pest management. Despite propagators' best efforts, small numbers of insects or mites are frequently present on cuttings received by growers. In particular, the whitefly, Bemisia tabaci, is regularly found on poinsettia. Scouting by OMAFRA over several years in Ontario greenhouses shows that infestation levels vary by year. For example, 2012 and 18 stand out as years with high whitefly pressure. Whiteflies mostly come in as eggs or small nymphs, and infestations are observed on many cultivars. Bemisia whiteflies are especially tricky because they have a pesticide-resistant biotype, which often represents the majority of whiteflies coming in on the cuttings. In 2018, we also did extensive monitoring of chrysanthemum cuttings. 84% of samples were infested with thrips. We found about 1 to 2 thrips per sample of 20 cuttings, mostly eggs and larvae. There was little variation among cultivars and time of year. Our goal was to look for thrips, but we also found that almost half of the chrysanthemum samples had 2 spotted spider mites. Numbers ranged from a few to over 100 mites of all developmental stages per sample of 20 cuttings. Infestations were quite variable and inconsistent, making this more of a hit or miss situation. So why is this important? Well, from a few pests here and there at the start of the crop, populations will inevitably grow rapidly. In this graph, you see that one can go from a few thrips to 6,000 in just 40 days. This head start for the pests makes it difficult for the bios to catch up, especially if pesticide residues on the cuttings, from sprays at the propagator, are affecting their performance. This would be a typical situation where a grower would consider to use a pesticide. However, pesticides may not work because the pests already developed resistance due to repeated exposure, and many pesticide options disrupt biocontrol programs for the target pests or for other pests, causing them to become problematic. Luckily, there are many things you can do to nip these pest problems in the bud, or in the cutting in this case. If possible, scout the incoming plant material so you have as much advance warning of the potential problems as you can. Unfortunately, you are looking for small numbers of tiny little bugs, so it may be easier to just assume that the plant material will be infested. Experience or records from previous years may support this assumption. Early intervention strategies include dipping cuttings, front-loading of bios, and the use of soft pesticides or biopesticides that will support your biocontrol program. In the rest of this presentation, I will talk more about cutting dips, in particular dipping unrooted cuttings. To do cutting dips, you take the cuttings out of the bag, spread them loosely in a perforated tray, cover this tray with a second tray, and completely immerse the trays in the dip solution. For small quantities or tiny cuttings, some people prefer to use a colander. The goal is really to get complete coverage of the cuttings. The products we investigated as dips were biopesticides or reduced risk products such as oils and soap. These products have less issues with pesticide resistance, leave minimal residues, and are highly compatible with biocontrol agents. After the cuttings are dipped, they are stuck in the substrate and grown as usual. When we started this research, there were many unknowns. We didn't know what the best products and rates were with regards to efficacy and phytotoxicity. Are the dips really compatible with biocontrol? And if it works in the lab, will it also work in commercial greenhouses? Another question was, how high is the risk of transmitting diseases through the dip suspension? Does dipping make sense from a cost-benefit perspective? So let's have a look at what we found. First, we had to find effective products and rates that would kill the target pest while not killing the plant. We tested Bemisia whiteflies on poinsettia, western flower thrips on chrysanthemums, and various bedding plants. And as we found that many cuttings were infested with both thrips and spider mites, we did some additional efficacy tests for spider mites on beans. These are just as easy as to grow, and spider mites love them. As you can see, we tested a lot of different products and rates. In red are the different oils. Blue is insecticidal soap, and entomopathogens, mostly fungi and nematodes, are indicated in green. We also tested many combinations of products. This table summarizes the most important results. You can see immediately that the different plant species could either be classified as hardy or sensitive, based on the phytotoxic effects of the products and rates. For example, the rates of soap or oil had to be lowered significantly in order not to damage the sensitive plant species such as poinsettia, mini roses, and many others. This is indicated by the yellow warning triangle with a plant that has no leaves. Luckily for each pest and plant combination, we did find a product and a rate that was effective. These are the dark green cells. In general, we saw mortality between 70 and 95%. Just note that the high rate of Botanigard was very effective against thrips, but only in the greenhouse trials that I will show later. That's why I only gave a qualitative rating for this product. A few more takeaways from these trials. First of all, it's very important to note that the dip rates of soap and oil are much lower than spray rates to prevent phytotoxicity. If you do it yourself, always test a small batch of cuttings for phytotoxicity first. The species and varieties you grow may be more sensitive than the ones we tested, and every greenhouse is different. We were happy to see that mineral oil like soft oil, X, and landscape oil also killed thrips' eggs. These eggs are inserted in the leaf tissue, making them very hard to kill. Finally, there were some products that were not effective as dips. For vegol, for example, we did not succeed in finding an effective rate that was not phytotoxic. Also, entomopathogenic nematodes did not do well as dips for unrooted cuttings. They may be more effective as dips for rooted cuttings, where they can accumulate and survive in the substrate around the roots. Next, we wanted to demonstrate the compatibility and the efficacy of a strategy where cuttings are dipped, followed by the release of biocontrol agents. We did this both for Bemisia whiteflies on poinsettia and for western flower thrips on chrysanthemums. For whiteflies, we dipped the cuttings in a combination of Copa insecticidal soap and Botanigard. For thrips, cuttings got either dipped in a high rate of Botanigard or in landscape oil. After the cuttings were dipped, or not dipped in the case of the control treatment, they were stuck in pots and arranged in plots of 24 or 30 pots on the mist. Releases of biocontrol agents were started immediately and repeated every week. Note that we did not include a treatment with no dips and no bios. I think we can safely assume that whitefly populations would have increased out of control rapidly. With biocontrol alone, which is the dotted blue line, we can see that whitefly numbers increased steadily. The dip knocked back the whitefly populations, as you can see from the solid yellow line, but without any follow-up control strategy, whitefly numbers started to increase again. Dips followed by biocontrol resulted in the best whitefly control, which is shown by the dotted yellow line. We obtained very similar results when we used Amblysia swirskii as the biocontrol agent instead of the parasitoids. For thrips, we basically see the same graph. With no dip and no bios, thrips populations increased rapidly, as you can see from the solid blue line. Using bios only slowed down this increase, which is the dotted blue line. Both the landscape oil dip and the Botanigard dip reduced thrips populations. The solid red and yellow lines are overlapping here. Dips and biocontrol combined resulted in the best thrips control, as represented by the dotted red and yellow lines. The thrips greenhouse trial turned into a spider mite trial because the chrysanthemum cuttings came in infested with spider mites. However, this allowed us to see some interesting results. Turns out that the landscape oil dip, the solid yellow line, also reduced spider mite populations, while Botanigard, the solid red line, did not. Amblysseus swerskii preyed on spider mites quite effectively, as you can see from the dotted red and yellow lines, but they were less effective in the treatment without dips, the dotted blue line, as they preferred to prey on thrips instead of spider mites when both were present. Despite these convincing data, there were still a few questions that needed to be answered before we could recommend dipping as a standard practice. Several growers and researchers expressed the concern for disease transfer from infected cuttings to healthy cuttings through the dip. For poinsettia, the main concern was soft rot-causing bacteria such as Owinia or Pectobacterium. This table shows the presence of these bacteria in different greenhouses, indicated by A, B, C, and D for 2 different weeks. We sampled the source water that was used to prepare the dip and found nothing. However, it seems that some of the dipping tanks were not cleaned properly. In greenhouse C and D, soft rot-causing bacteria were found in quite high numbers, even before dipping any cuttings. During and after cutting dips, these numbers increased up to levels of 10 to the 5th, which is 100,000 colony-forming units, CFUs, per milliliter. It is important to notice that this kind of bacteria can be found on many surfaces in the greenhouse, not only on cuttings, so good sanitation practices will help prevent accumulation of these bacteria. It's also interesting to note that despite the higher levels of bacteria in the dips, no disease was observed in the dipped cuttings afterwards. We did also an experiment to see if these levels of bacteria in the dip solution could actually infect healthy cuttings. So we prepared dips with increasing concentrations of Pectobacterium from very little, like 10 to the 3rd, to a Pectobacterium soup at 10 to the 7th. The green arrow indicates the middle concentration that was found in the commercial greenhouse dips, as from the previous slide. We also included clean water, cuttings injected with Pectobacterium as a positive control, and non-dipped clean cuttings as a negative control. 2 different varieties were tested, namely Prestige Red, which is a more susceptible variety, and Freedom White, which is a less susceptible variety. Each treatment was applied to groups of 20 cuttings, and everything was replicated and repeated. We can see immediately that Prestige Red, the light bars, developed more disease than Freedom White, the dark bars. Even some of the non-dipped cuttings and cuttings dipped in clean water developed disease, as you can see on the left of the graph. Second, disease incidence increased with increasing Pectobacterium concentrations in the dip. However, a statistically significant difference was only observed at the highest concentration of 10 to the 7th CFUs per milliliters. In other words, this was the only treatment when more disease was found as compared to the other treatments, including the non-dipped control that were all the same. On the far right, most cuttings injected with Pectobacterium developed disease, showing that we did use an infective strain of bacteria. This experiment demonstrated that the risk of Pectobacterium transfer among unrooted cuttings in the dip is low. There's no time to go into details, but we also did a cost-benefit analysis comparing biocontrol alone, dips combined with biocontrol, and a chemical control option. Based on the cost of product, labor, losses due to pests, and total value of the crop at sale, we found that all three strategies yielded net benefits that were very close to each other. DIPs and biocontrol together yielded slightly higher net benefits than biocontrol alone. Chemical control yielded the highest net benefits, but this calculation did not take into account the risk of pesticide resistance. In general, DIPs can be viewed as a small insurance premium to reduce risk. During bad whitefly years, it really pays off. In years without whiteflies, DIPs do not add many additional costs. In conclusion, we found that DIPs can effectively suppress whiteflies, thrips, and spider mites. DIPs are compatible with biocontrol agents and improve the overall biocontrol program. The risk of disease transfer is low in poinsettia, but research did emphasize the importance of good sanitation practices. Finally, the cost-benefit analysis showed that DIPs make sense economically. As a result of this research, dipping has been added to the product labels as an application method. Especially in Canada, there are now 4 products that can be used as dips. The uptake by growers has been great, and dips have helped mitigate high whitefly pressure on poinsettia, for example, in 2018. Of course, we don't have all the answers yet. There are many opportunities for further research, including more products, more pests, and more crops, and also automation of the dipping process and integration with production practices. For your reference, we made a short video explaining all the important steps of cutting dips. You can find it following this link on the screen. It's also on the Greenhouse Canada and Vineland Research and Innovation Centre websites. I'd like to end with a few pointers on how to test dips yourself, especially to test for phytotoxicity on a new crop. First of all, don't go in with your entire crop. Test a few smaller samples first. For cuttings, we recommend samples of 10 to 20 cuttings. Second, replication is important to make sure the results are not due to chance alone. Do at least 3 samples of 10 to 20 cuttings per product or per rate. Also, do not assume it's okay for one variety, that it's then okay for all varieties. Finally, factors like environmental conditions change over time, so repeat the test at different time points. Third, make sure to include a non-treated control, either dipped in clear water or not dipped, so you have something to compare to. Finally, quantify the results. This could be leaf surface area affected or a damage scale. Although you're probably not going to publish the results in a scientific journal, just eyeballing the plants won't convince anyone. You have to quantify. Of course, you can always enlist our help or the help of an Omafra specialist. I would really like to thank all the people and companies that helped with the research on this slide and on the next slide for their hard work, their support, and their involvement with this project. If you have any questions, here is my contact information. I'll be happy to help, and thank you for listening.